All right. Up next, Michelle Yee.
I'm Michelle from the NYU Health Sciences Library. A lot of our librarians are here, and it's great to see you. I am presenting this on behalf of a team that's been working on the NIH Brain Initiative. Now, this is a very broad study under the neurosciences. Well, let me start from the beginning. This is a very broad study launched by the National Institutes of Health in 2014 to develop effectively a research network, many hubs across the US to study systems neuroscience, development of neuroimaging technologies, and how to implement data science and informatics infrastructure in this research area. At NYU Langone, this project is led by Dr. Tian, Dr. Bishaki, Drs. Franke, and Lin. All of the funded programs through this NIH Brain Initiative are required to establish a data science core. And this data science core at NYU is led by Alisa Surkis. All of the data science cores under the Brain Initiative form this data science consortium. And this consortium has been tasked to tackle integration, interoperability, and data reuse challenges from the perspectives of investigators at each data science core. Now, this is significant because the investigators themselves represent not only their institutions, but all of the labs that work with them at the institution. So we're talking about ideas from hundreds of scientists effectively. So out of this data science consortium, two teams, one here at NYU, as well as the Allen Institute for Neurodynamics, had found that we both independently had come up with platforms for capturing information about neuroscience experiments. Here at NYU, this tool is called Brainstem, and I'll speak a little more about this in some of the upcoming slides. But because we were taking such similar approaches, we decided to coordinate our approaches for describing neuroscience experiments in order to maximize reproducibility. We would effectively look for similarities and differences between the metadata schema that we had developed for our respective tools, and also review the underlying logic for each schema. So just a little bit of a vocabulary check. Metadata, this is information that provides a description or classifies or contextualizes another resource. So here we have some descriptions related to experiments, or descriptors related to experiments. These include subject species, the age, weight, things like that. Organizing this metadata into a schema. A schema is effectively an outline that describes how this information is organized. So species, age, and weight, these are the characteristics of a subject, for example. And then finally, a data model is really a representation of how this metadata fits together, how they relate to one another. Formatting is a little funny, but you can get the idea of how, again, these are related. Data standards, the last thing I'll be defining. These are formally defined guidelines for describing, collecting, or structuring data in order to maximize how they can be used, reinterpreted, or exchanged. Dr. Bradella had talked about DICOM and its importance in the transfer of imaging. FHIR is another significant one for transmitting electronic health records. And some of you may be familiar with MinSeq if you work with genomics data, or CPT if you work with surgical, if you looked at surgical data. These standards are typically community defined to meet the needs of specific disciplines. In research, having data standards enables various analytical strategies like meta-analysis or machine learning. And because of this, standards are an ideal. If not, ideally, if not, an approach that we could take is interoperability. Within neuroscience research, two primary standards have emerged. These are Neurodata Without Borders for neurophysiological data, as well as the Brain Imaging Data Structure, or BIDS, standard for neuroimaging data. This figure to the right shows how the BID structure, what it looks like, how it organizes different file types, and specifically how EEG data streams can be integrated into the BID standard. So this is a screenshot from the brainstem. It is an electronic notebook similar to Benchling, if that is something people have used. The structured fields in brainstem make it easier for researchers to report out on various aspects of their projects. And all these fields can also, of course, be used as a reference when writing manuscripts or even completing a STAR reporting, STAR resources table. Here's a look at the brainstem data model. It builds upon the Neurodata Without Borders data standards for describing neurophysiological experiments. So it focuses on, the foundation is information that is important for measuring brain response and behavior to stimuli. So it captures, you can see that's reflected in the data acquisition and manipulations tables. Do keep in mind, while we move on, the first couple of classes in this table, it starts with projects, and beneath projects, how they're linked to subjects and sessions. This will be relevant when we look at the next one, which is the Allen Institute for Neurodynamics. And although they do not have a visual representation of their schema, this is something that was developed in alignment with the brain imaging data structure, and where brainstem has starts with a project and has subjects branching off that. The Allen Institute for Neurodynamics starts more flat. So what did we actually do? The two teams met to create a crosswalk. This crosswalk includes separate sheets for those high-level classes I mentioned earlier. So that would be subjects, procedures, and sessions. We had separate sheets for vocabulary related to these concepts. Each row contained a single concept or term, and our different teams' implementations were organized in separate columns. Similarities and differences were color-coded and discussed. So this green here is for terms that are conceptually the same, orange for concepts where one is missing in one of the schemas, and blue is also represents concepts that are missing in one schema, but after discussion, we felt were not critical to align. So the main general finding of this activity was that what we had thought of as differences that we had found during this crosswalk activities, they were due to underlying differences in research activities, infrastructure, and design approaches. So what does this mean? For example, drawing from local and centralized resources, the Allen Institute's for Neurodynamics schema had contained fields that pulled in information from their internal databases, whereas brainstem was developed with a more flexible approach in mind to accommodate distributed research systems, so labs that were managing their own resources. So lessons learned from this crosswalk activity, these may seem quite obvious, but I think are important to reflect on as we are developing tools and considering how we can make our data interoperable or tools interoperable. So first of all, remember that tools are built with implicit assumptions about their users. So in terms of our crosswalking exercise, it was really critical to have a discussion about the decisions that were made. The assumptions, again, about users were reflected in those structural differences that we had observed in the schema. And where consensus was not feasible, working towards interoperability was not really a compromise, but it was the most feasible path by finding areas that were already compatible rather than more divergent. We were able to make changes that supported data harmonization. So for example, while Yale Institute and Brainstem schemas didn't have the exact same naming system for individual subjects, we added to our respective brain atlases for describing the relative positions of surgeries, for example. And then finally, and this is a lesson I think that has been repeated across multiple presentations, but cross-disciplinary teams have been key. So our discussions involved not only researchers, but developers of the tools and data librarians. And through these discussions, we balanced what were discussions about ideals or best practices, what were the realities of research data management. So what actually made it easier for researchers, what would make it easier for researchers to record their studies. The ultimate goal really from these tools is to make it easier, deduplicate the effort needed to describe data collection, to communicate findings, and then ultimately share and discover each other's data sets. So I want to acknowledge our collaborators at the Allen Institute and the funding from the BRAIN Initiative. All right, and thank you.